Hello, my name is Aptin Riyasatian and I'm a master's student at the University of Waterloo Kimia Lab. I will present you the paper titled A Comparative Study of Unit Topologies for Background Removal in Histopathology Images, co-authored by Mara Rasooli Jaberi and Morteza Babai, supervised by Professor Hamid Tishush. In this presentation, first I will define the problem of background removal in histopathology images. Then I will talk about the importance, applications, and challenges of this problem. After doing a quick literature review, I'll talk about our data preparation and training methodology. And finally, I will go over the experiments and analysis of the results. As the name suggests, the goal in the problem of background removal or tissue segmentation in histopathology images is to remove irrelevant and uninformative pixels as much as possible, with the minimum damage to the tissue pixels. Also, since histopathology image analysis is generally the last step for cancer diagnosis, it is crucial to avoid losing tissue pixels. So, the sensitivity of the segmentation algorithm has more importance than its specificity. In many histopathology image analysis tasks, extracting the tissue parts is the first step. So, segmenting the tissue precisely is a prerequisite for an effective algorithm. On the other hand, if this is not done well, irrelevant parts will confuse the algorithm. Tissue segmentation can be used to speed up the digital pathology slide scanners. These scanners are used to digitize glass slides containing tissue specimens and generate WSIs, which stand for whole slide images. While scanning, the focus depth of the scanner must be adjusted for different tissue regions due to variable tissue thickness. So, scanners need to identify all tissue areas. If a mistake occurs during scanning, the data is lost and there is no way to retrieve it in the following steps of the workflow. Currently, a technician manually checks every slide after scanning and rescans the corrupted slides, which is a tedious and expensive procedure. Extracting the tissue parts of the slide before starting the scanning can reduce time and cost of this process while doing it at a higher precision. Generally, this task has two types of challenges. The first ones are related to the tissue type. For instance, as you can see, air sacs in the lung and fat, which could appear in many tissue types, may confuse algorithms due to their similarity to the background color. The other category is artifacts, which include extra or weak stain, dirt, bubbles, broken glass, and marker traces. There's not much research done on histopathology image background removal as a standalone problem, and it's often tackled as a part of a larger pipeline, but in general, there are two types of approaches to do this task. The first category is the machine vision-based methods, which include performing thresholding on features such as image saturation and intensity and applying the Otsu method on different color spaces. One of the competitive algorithms for this problem is the FESI algorithm by Bug and others, which uses a combination of basic methods such as median filtering, thresholding, erosion, and dilation to address this problem. Also, one of the most well-known and widely used open-source libraries in digital histopathology called Histomix Toolkit or Histomix TK provides a tissue detection function which contains a series of Gaussian smoothing and Otsu thresholding. For the learning-based methods, two research papers can be mentioned for this problem. Alamari and others have fed 4 features including color, appearance, texture, and spatial features to a 2-layer neural network to classify the pixels into background and foreground classes. Also, Bondi and others trained a fully convolutional neural network with 7 convolutional layers as well as a unit for tissue segmentation. The input for these networks were patches with a single label of tissue or background based on their central pixel. Our main contributions in this paper are Creating a manually labeled dataset consisting of 244 thumbnails chosen from the publicly available TCGA dataset, which stands for the Cancer Genome Atlas, as well as comparing the performance of different encoders as the backbone of unit topology for the tissue segmentation task, and proposing a tailored algorithm for this problem. To create the dataset, we used 244 WSIs from different organs such as brain, breast, kidney, and lung. All WSIs were randomly selected from the TCGA dataset. One of the challenges of histopathology image analysis is the WSI size, which could be as large as 100,000 by 100,000 pixels. This means that labeling each pixel is not practical. To overcome this problem, we chose to work with the thumbnails, which are generally around 1x magnification. In addition to easier labeling, this has the advantage of fast computation. Creating tissue masks was performed in 2.5x magnification, as we found it to be the lowest magnification that lets us distinguish the tissue parts from artifacts such as pore staining. Labeling process consists of 3 steps. First, we developed a handcrafted algorithm to create initial mask for every thumbnail. This algorithm applies binary thresholding on the grayscale thumbnails and does some processing based on the size of contours and their distances. After that, the initial masks were refined manually to make sure all tissue regions are selected and noise and artifacts are removed as much as possible. At this step, dilation was performed on difficult cases to make sure all tissue parts are preserved. Finally, each pair of mask and thumbnail is resized to make each dimension less than 1024 pixels to make the images small enough to be fed to the network. We chose UNIT as the general topology due to its impressive performance in various medical imaging segmentation tasks. Different networks were compared by utilizing feature extractors of architectures such as MobileNet and EfficientNet-B3 as the encoder part of the unit. The decoder part was fixed and made up of 5 blocks of upsampling plus convolutional batch normalization and ReLU layers. For the training process, the networks were initialized with ImageNet weights and trained for 50 epochs with no early stopping using Atom Optimizer. For augmenting the input images, rotation with random angle, random horizontal flipping, and random vertical flipping were used. We used sensitivity plus specificity as a loss function, so the network tries to come up with an approximation which avoids missing the tissue parts while having a good performance at recognizing the background pixels. We did experiments with 6 different backbones, including MobileNet, EfficientNet-B3, ResNet50, DenseNet121, ResNext101, and VGG16. The performance of the networks were evaluated using 5-fold cross-validation. In order to compare our results against available established methods, we also tested algorithms such as OTSU, TissueLock, HistomixTK, and an improved version of FESI on our dataset. Comparing the network's results to the current well-known methods, it can be observed that all networks except VGG16 have outperformed these methods according to their Jaccard index. While the best sensitivity among the available methods is 91%, for FESI, the sensitivity of all networks is 99%. This is the most important factor in this problem and means that our algorithms have managed to minimize missing the tissue parts. Also, the specificity for the networks except VGG16 is equal or higher than 98%. Finally, MobileNet and EfficientNet-B3 have outperformed all algorithms with specificity of 99% and 98% respectively, while maintaining a 99% sensitivity. It is worth mentioning that the reason behind the low performance of VGG16 could be due to lack of batch normalization and ReLU activation layers, and the fact that it's rather a wide network than a deep one. The manifest to download the data from the GDC website, the dataset used in this research, as well as the codes and weights to run the proposed networks will be soon available for download at the Kimia Lab website. I appreciate your time for listening to this presentation.